Welcome to The Explainer. If you're the kind of person who loves getting straight to the fascinating core of how our world works, well, you're in the right place. Today, we're diving into the staggering and honestly almost unbelievable journey that a single chemical has to take just to become a medicine. We call this the gauntlet, from 10,000 compounds to one cure. Let's get right into it.
10,000. Take just a second to let that number really sink in. Because the process of discovering a new drug, it doesn't start with just a couple of bright ideas scribbled on a lab whiteboard. No, it kicks off with a massive, sweeping search through an initial pool of 10,000 different chemical compounds.
And look, if sorting through 10,000 compounds sounds like a monumental task, get a load of the timeline. On average, it takes a grueling 10 to 15 years of relentless science, testing, and strict regulatory review to get just one new drug onto the market. 15 years! I mean, think about that. That is a massive chunk of a scientist's entire career dedicated to just one single potential cure.
But before we get ahead of ourselves, let's just ground things with a simple definition. What exactly are we trying to build here? Well, at its core, a drug is any chemical that produces a change in the body. But the absolute key word here is selectively. It has to be selectively used in the diagnosis or treatment of a disease. It's that selectivity, the ability to target the bad stuff without destroying the good stuff, that makes this whole process so incredibly tough. So why do we even do it? Why take on a 15-year gauntlet with such astronomical odds?
That brings us to Chapter 1, the quest for cures, and why we desperately need new drug discovery.
Basically, we are facing a massive societal burden, and it's not just about battling incredibly complex diseases like Alzheimer's or the rising rates of obesity. It's also the reality that many of the treatments we already have, like certain cancer therapies or dementia treatments, honestly just have low efficacy. Or think about medications like antipsychotics and antidepressants. They can come with severe, sometimes debilitating side effects. And without developing new therapies, the downstream health costs for conditions like spinal injuries or severe depression are just devastating, both to the individual and to the broader economy.
And just look at the sheer complexity of what we're up against here. These intricate cellular networks, like neurons in a brain affected by Alzheimer's. This is what scientists are trying to fix. They aren't just mixing some random chemicals in a beaker. They're trying to correct incredibly complex, microscopic cellular and genetic misfires. It's literally like trying to repair a supercomputer using a wrench. That is exactly why we need highly specific, perfectly targeted drugs. But testing these chemical wrenches on actual human beings, obviously that's incredibly risky.
Which leads us to Chapter 2, a history of human trials and the evolution of safety and ethics.
You see, we didn't always have the strict, rigorous rules we rely on today. Way back in 1537, Ambroise Paré ran what's essentially considered the first clinical trial of a novel therapy. Fast forward to 1747, and you've got James Lind famously using control groups to prove that citrus fruits actually prevent scurvy. Then placebos pop up around 1863. But it wasn't until the horrors of the mid-20th century that things really fundamentally changed. By 1945, strict regulations on human medical experiments started taking shape. Those rules were firmly enshrined in the Nuremberg Codex in 1947 and later followed by the landmark Declaration of Helsinki in 1964. And today, those ethical guidelines are the absolute foundation for everything we do.
All right, Chapter 3, the 10,000 compound funnel, surviving the ultimate obstacle course.
The sheer scale of failure we're talking about here is just staggering. Watch how dramatically this funnel narrows down. We start way over on the left with our 10,000 initial compounds, but by the time you push through basic discovery and into preclinical testing, that huge number is whittled all the way down to just 250. It is a massive, unforgiving attrition funnel, specifically designed to ruthlessly eliminate absolutely anything that isn't safe and effective. To actually survive this funnel, every single compound has to navigate three main pathways. First up is discovery, which is all about identification and picking the right target. Second is preclinical development, where we rely heavily on animal and safety studies. And finally, clinical development. That's the big one, human testing and ultimately the push for FDA approval.
So, during that first discovery phase, scientists use a process called combinatorial chemistry. Using these high-throughput screening plates, fully automated robots rapidly test thousands of compounds against a highly specific cellular or genetic target, just trying to isolate potential leads. It's incredibly fast, and it systematically searches for that one tiny needle in a giant chemical haystack.
Now, if a compound is actually lucky enough to be among the 250 that make it out of discovery, it moves into preclinical development. Before a chemical ever gets anywhere near a human being, it spends roughly a year and a half undergoing rigorous studies. Scientists use animal models of disease states to study pharmacokinetics, basically figuring out how the body absorbs, distributes, and processes the drug. They also run short-term toxicology studies just to make absolutely sure it isn't outright poisonous.
Moving on to Chapter 4, the clinical trial gauntlet, efficacy, dosage, and the ultimate test.
When we follow the clinical development roadmap, things get really real. Out of those initial 10,000 compounds we started with, only about 5, yes, literally 5 development compounds even make it to phase 1 of human clinical trials. The odds are just wild.
And when those 5 final compounds enter phase 1, scientists aren't even asking if the drug cures the disease yet. They are zeroed in on one simple, absolutely vital question: is it safe?
Phase I is focused purely on pharmacokinetics, tolerability, and side effects. This is done with a very small group of healthy volunteers. The entire goal here is simply to guarantee that this completely novel chemical won't harm a healthy human body. Because if it causes severe toxicity at this stage, that's it. The journey is entirely over.
But if the compound is proven safe in healthy people, we finally get to ask the question we've literally been waiting years to ask. Does it work?
Answering that question is what Phase 2 is all about. This involves small-scale trials in actual patients who have the target disease. Here, scientists are trying to assess efficacy, figure out the exact proper dosage, and look really closely at long-term toxicology. And during this phase, the candidate pool shrinks even more, usually down to just 3 surviving compounds.
Okay, now we arrive at the ultimate scientific test. The one question that makes or breaks a pharmaceutical company's massive multi-million-dollar investment. Does it work in double-blind trials?
Welcome to the incredibly high-stakes world of Phase 3. This stage requires large-scale, highly controlled clinical trials across hundreds or even thousands of patients. The goal is to definitively prove medical utility compared to a placebo or to an already existing treatment. This massive final test is unbelievably expensive and rigorous, and it essentially narrows the field down from about 1.7 compounds right to the finish line.
And here it is, the sole survivor of the funnel. Out of 10,000 initial compounds, after 15 years of endless discovery, animal testing, and 3 grueling phases of human trials, we are finally left with exactly one FDA-approved drug ready for marketing. I mean, it is truly a monumental achievement of human ingenuity and sheer perseverance.
But here's the thing. The story actually doesn't end there. We still have Phase 4. Post-marketing surveillance ensures indefinite safety follow-ups, even after the drug hits the market. Because sometimes extremely rare side effects only become apparent when millions of people are actually taking the medication in the real world. So the monitoring, it never truly stops.
So I want to leave you with this final thought. The next time you twist open a bottle and take a simple pill for a headache or maybe an infection, will you think about the 10,000 failures and the 15 years of relentless, grinding science it took to get that medicine to your cabinet? It really gives you a profound appreciation for what goes into drug discovery, doesn't it? Thanks for joining me on this explainer. The next time you open your medicine cabinet, you'll know exactly what it took to fill it.